So, welcome back, all of you, to the online class of biology. Today, we are continuing the topic DNA fingerprinting. Okay, so in the last video, we have started the topic and I explained you some terms related to this, like repetitive DNA, satellite DNA, then what microsatellite, mini satellite, etc. Okay, so do you remember what is repetitive DNA? That is, repetitive DNA includes what? DNA sequence or nitrogen based sequence which repeats many times. Okay, so DNA fingerprinting, they mainly focus on this repeated repetitive DNA. Okay, because as we have studied Human Genome Project, it was a very tedious what project or it is work. Okay, it is very costly and it needs what? The hard work of many scientists for many years with the expenditure of lot of money. So every time to compare the DNA of two persons, it is not possible. So in DNA fingerprinting, what focus is done is in some specific regions of DNA that are called as what? Repetitive DNA. So I told in repetitive DNA what are the different types. Okay, like what? Mini satellite, micro satellites, depends upon what the number of repeating units, isn't it? How many times they are repeated. So on all that basis, they are classified into micro and mini satellites. Okay, and I also told you about the polymorphism. What do you mean by polymorphism? Difference. Okay, differences that mainly occur or due to what? The mutations. Okay, so now I'm going to say about the person behind this. He is Alec Jeffries. Okay, so the DNA fingerprinting technology was first developed by Alec Jeffries. Okay, Alec Jeffries. So Alec Jeffries found out, okay, or developed this technique, okay, DNA fingerprinting technique, and he used some repetitive DNA or satellite DNA, okay, which showed very high degree of polymorphism, and it was called as what? VNTRs. Okay, so what? How can you define VNTRs? VNTRs are, okay, they are satellite DNA. What are satellite DNA? Which will get separated as what? Mini satellite and what? Bulk DNA, isn't it? So the what? The minor peaks that we get when we do density gradient centrifugation. So that are what? Satellite DNA, which contains what? Repetitive DNA. Many times the nitrogen bases will be repeated. So this technique was developed by Alec Jeffries. Okay, Alec Jeffries, and he used what? VNTRs. What are VNTRs? Variable number of tandem repeats. Means satellite DNA with high degree of polymorphism he used. Okay, now I'm going to teach you about the different steps involved in DNA fingerprinting. Okay, so the first step that includes involved is isolation of DNA. So from, if it is what the DNA fingerprinting is done in a to identify the criminal of a particular crime, then from the crime scene we get some samples of cells, like what? How much ever the criminals try to what? Hide it. Okay, some cells, okay, of blood it can be, or semen, or it can be of what? Of air follicles. All this what evidences they'll leave there, isn't it? So the from this evidences, so the policemen collect the samples, and from there first what they have to do is they have to isolate the DNA. Okay, so isolate the DNA. So first step is what? Isolation of DNA. Okay, then next step is what? Digestion of DNA by restriction endonucleases. Okay, so restriction endonucleases you have not studied. In the biotechnology chapter we will study. So these are enzymes which can cut the DNA in such a way that, okay, they produce certain sticky ends. Okay, we have studied, isn't it, palindromic sequences, and also likewise, okay, they can cut the DNA. Okay, so DNA will be cut into the isolated DNA will be converted into a number of small fragments. Okay, now next technique or next step is separation of DNA fragments by electrophoresis. So this you might not have studied. That is, there will be a agarose gel matrix. Okay, gel matrix will be there, and into the one side of the matrix, okay, gel electrophoresis will be what, having a gel matrix in them, agarose gel mainly, and this into this agarose gel medium we will put the DNA sample. Okay, as DNA being negatively charged particles, they will move towards the positively charged what, electrode, isn't it? So when they move like that, the smaller particles will move fast, and the larger particles will get what, they will move slow back. Understood? So likewise, what, according to their size, the DNA molecules will start moving. Okay, according to their size and charge. Then next, so that is gel electrophoresis is done for what? For the separation of DNA fragments. Now next step is what, the separated DNA fragments. Okay, so that has to be copied into certain synthetic membranes such as nitrocellulose, nylon, etc. Okay, so first what? Isolation of DNA, then the DNA will be cut into smaller pieces. Then what? This pieces all together will be put into gel electrophoresis, whose DNA move, then this DNA will move, okay, towards the positive electrode, isn't it, as the DNA is negatively charged molecules. Then what happened? This will be, DNA will be in the agarose gel in different parts. Why? Because their charge, their size is different, so they will be getting separated into different parts. Now next step is what? Transferring the separated DNA fragments into some nitrocellulose or nylon what, membranes. So for this, from the, now the DNA will be where? In the agarose gel medium. So it has to be what, transferred to a what, synthetic membrane, like what, nylon and nitrocellulose. To that it will be copied. Okay, then next is what? Detection of hybridized DNA by autoradiography. Then we use what? VNTRs. Okay, understood? Then what? Hybridization using labeled VNTR probe. Okay, so non VNTR probes are used to hybridize with the DNA. Okay, so when they what, hybridize, okay, in which part of the DNA they get hybridized, okay, that can be detected by autoradiography. Okay, so nowadays hybridization is also using labeled VNDR probes. Okay, so that is also possible. Now what we have to do is to compare the DNA. So these are the steps of DNA fingerprinting, that is isolation of DNA, digestion of DNA by restriction nucleases, separation of DNA fragments by gel electrophoresis. Then what for gel electrophoresis is done? For separating the different DNA fragments. Then these DNA fragments will be what, will be transferred to nitrocellulose or nylon membranes. Then next step is what? This hybridization of DNA with the VNTR probes, which are already known. Okay, then this VNTR probes will go and bind to the DNA. Okay, and form what? Hybridized DNA fragments. And This can be what observed, okay, by auto radiography. So these are the steps used in where in DNA fingerprinting. Okay, so the VNTRs are kind of mini satellites, and they occur in many copy numbers, means many times it will be occurring. Okay, and this copy number, how many times it repeats from chromosome to chromosome, okay, in an individual, and varies from one individual to another individual or from chromosome to chromosome. Okay, and the number of repeat, means how many times it repeats, will be entirely different from one person to another. Okay, so the size of the VNTR can be from zero point 0.1 to 20 kb, means it varies. In some of them it will be very less, okay, repeating very less, or in some others they'll be repeating more. So likewise what this VNTRs will be different, okay, their repeating units will be different. So then what happened is, as they are hybridized with the DNA, then what happens, auto radiography we do. Okay, so that what can be done, this bands, okay, wherever the DNA hybridization happened, okay, the DNA VNTR probe will go and bind to the DNA, isn't it? So wherever it happened there, we can see what bands be, that different bands we can see. Okay, so this bands, that will be very specific to a person. Okay, means in the DNA obtained from any samples, like it can be blood, semen, or air follicles. In all this, the the DNA found will be same with the same polymorphism. Is it clear? Or the bands will be what very similar, okay, in all the cells of a particular individual, and it will be different from others DNA. Understood? So this bands which are formed will be what it will be checked by what the checked for comparing the DNA. Okay, so here we can see one chart. So this is a schematic representation of DNA fingerprinting. Okay, so few representative chromosomes have been shown to contain different copy numbers of VNTR. You can see, okay, the first one is chromosome 7, okay, chromosome 2, and the last one is chromosome 10. Okay, so you can see that this are some of the chromosomes. Okay, they are having what VNTRs, you can see. Okay, different colors are used here. See, in paternal, this is the paternal what chromosome, and this is the maternal one. In an individual, this both will be there, isn't it? So in an individual you can find what, two, one from the mother and the other will be from the father. So if you compare this two, you can see that there is difference. And likewise only if you compare what, this two, okay, the this two, there is what difference. Okay, so VNTRs will be varying in chromosomes to chromosome and from individuals to individual, and this yellow one is showing another one. Okay, then likewise what different VNTRs are shown. Okay, then so same pattern you will get where also, okay, in the DNA fingerprinting, isn't it? So after doing this DNA fingerprinting, you can see this is the what DNA fingerprinting done from the sample which we obtained from the crime scene. Okay, so after doing this, okay, you can see here this what DNAs, Okay, a fingerprint that will be see like this. This is the what one which we get from the okay crime scene. Okay, and when we do the DNA fingerprinting, we get the bands like this. Okay, and this are from two individuals. Okay, this are from the two individuals. One is from here, the other is here. Okay, now you compare the DNA patterns. You can see that C and B, they are exactly similar, and the what A. Okay, A is what, that A and B were suspects, means they were doubted to be done the crime. So when we compare the DNA fingerprinting of the sample obtained from the crime scene and of the suspects, we can see that the criminal who had done the crime and that of the DNA which was collected from the crime scene will be showing the same pattern of arrangement. Okay, you can see that A and C, B and C. Okay, and A will be entirely different. So one more chance is there, that is the between the relatives, blood relatives. Okay, the chance of the similarity will be more. If as the relation between two individuals are very close, the chance of this bands also will be to become what same will be more than two unrelated regions. So this is the what use of this DNA fingerprinting in paternity test. What is paternity test? To understand the means to confirm a person as what one's father. Understood? So for that also it can be used. Okay, so this is what DNA fingerprint. From this it is very clear that what VNDRs shows what different what repeating units, or they are having different repeating units. Then what they are used for what comparing. Okay, so in DNA fingerprinting, this is the DNA fingerprint that obtained from the sample of a crime scene, and these are from two different individuals. Okay, so the what A, sorry B site or B's DNA fingerprint was more matching to C, confirming that what B has done the crime. Okay, so it can be used for what also, for identifying the what paternity, or for paternity test to identify a person's father. Okay, so between blood relatives, the chance of this band to be more similar will be more than what two different individuals. So it can be used for identifying the paternity. Understood? Okay, so in a technique known as PCR, polymerase chain reaction, are used to amplify the DNA, because from the crime scene, okay, the DNA obtained might be very less. So the obtained DNA every time to isolate and all, it is very difficult. So the obtained DNA can be amplified using PCR. So what are the different use? Determining the population and genetic diversity, it can be used. Okay, then in forensic sciences, in case of paternity disputes, in all these fields, DNA fingerprinting is used. Is it clear to everyone what is DNA fingerprinting and the various steps to be followed in the process of what the DNA fingerprinting? Is it clear to everyone?